Hello grade 11 students. In this video we're going to talk about our new lessons about transgenesis and the process of transcription and transgenesis. What's the meaning of transgenesis, or what is transgenesis? Transgenesis is the process where a gene is transferred from one species to another. Pay attention to this. From one species to another, and it's expressed in the other species. For example, I transfer a gene from human to a mouse, from a human to a cat, from a cat to a donkey, from a fish to, for example, to a lizard, let's say. Okay, so this process is called transgenesis. So transgenesis, it's the process by which a gene is transferred from one species to another, and this gene should be expressed in the later. This later is called the transgenic. So whenever I transfer a gene from a human to a mouse, and the gene is expressed in the mouse, this is a human gene. So this mouse is called to be transgenic mouse, since it carries a gene from another species. So first of all, you have to know what is transgenesis. Transgenesis is the process where a gene is transferred from one species to another and should be expressed in the later. Also, you have to know the meaning of transgenic. Transgenic is an organism that carries and expresses a gene from another species and should be expressed in. Let's move to this experiment. In this experiment, we have removed eggs from a mouse, and then we inject the human insulin gene in the form of DNA in the male pronucleus, and then the male pronucleus is injected into the female pronucleus, and we have the embryos where they're transferred into the uterus of a foster mother. So I repeat, we remove the eggs from the mouse, we inject the human insulin gene. The human insulin gene means that the insulin gene in human, we inject it into the mouse, and we inject it in the form of DNA in the male pronucleus. This male pronucleus is injected into the female, and we have the result, the embryos. So transfer of the number of altered embryos into the uterus of the foster mother. What was the result? The result was the birth of 3 mice. 2 of them, they showed a negative result, meaning that there is no insertion of the DNA fragment, meaning that there is no insertion of the human insulin gene in the form of DNA, while 1 shows a positive result, meaning that it shows the human insulin gene in the mouse. So this mouse is said to be transgenic. Why? Because it expresses a gene from another species. So let's describe. Describe, as I said before, it should be in the form of passive and should not include any additional information. So the eggs are removed from the mouse, then the human insulin gene in the form of DNA is injected into the male mouse pronucleus, which is then injected into the female pronucleus. Then a number of altered embryos, they are transferred into the uterus of the foster mother. Then after the birth of 3 mice and search for human insulin gene, there are 2 mice, they showed negative result where there is no insertion of the DNA fragment, but 1 mouse showed a positive result, and this mouse is classified as transgenic. Interpret. It's analysis plus indication. So after injection of a male pronucleus of a mouse previously injected by the human insulin gene into the form of DNA into female mouse pronucleus and transferring the resulting altered embryos into the foster mother, the result was the birth of 2 mice with negative result and 1 transgenic mice with positive. What does this indicate? This indicates that the human insulin gene in the form of DNA could code for the production of human insulin hormone in the transgenic mice. Conclusion. So a gene is made up of DNA, can code for the production of specific proteins. Chromosomes, we talk about this, they are at metaphase, they are made of 2 sister chromatids. The substance that make the chromosome is called chromatin. Chromatin is form of DNA composed of nucleotides A, T, C, and G. They are the 4 nucleotides that are forming the DNA. A, T, C, and G, they are called the nucleotides. Every 3 together, for example, we have ATC. Okay, ATC, they're composed of 3. It's called triplet, or I'm going to use a new word this here is called codon. C-O-D-O-N. It's called codon. So every 3 together, they are called codon, or they have the name triplet. Okay? You have to know that DNA is double-stranded. DNA is called deoxyribonucleic acid. DNA is composed of the nucleotide A, T, C, G, and A is complementary to the T. C is complementary to the G. So A and T, they're complementary. For example, here we have two strands. In this strand, we have the A. In the second strand, we should have T, because they are complementary. In the first strand, we have G. The second should be C. In the first strand, we have A, so it should be T. The first strand, we have G. The second should be C, and so on. This is the sequence of the DNA, where A and T, they are complementary. C and G, they are complementary. Also, we have proteins composed of histones and non-histones. We're going to talk about them later. But now, we will focus on the nucleotides A, T, C, and G, and we will work on this whenever we talk about transcription and translation. Here we go. DNA, by the process of transcription, will give mRNA, messenger ribonucleic acid. DNA is called deoxyribonucleic acid. And mRNA, by the process of translation, will give polypeptide, will give amino acid, chain of amino acids called polypeptide. So what is transcription? Transcription is the process where the mRNA is produced based on the sequence of the DNA. Mainly, this process, it occurs into the nucleus. What is the fate of produced mRNA? The produced mRNA undergo mutation, and we will talk about mutation next week, inshallah. So, and they are transported from the nucleus to the cytoplasm. So what happens? DNA, by the process of transcription, will form what's known by mRNA. mRNA is formed under the action of two enzymes, helicase and RNA polymerase. What is the action of helicase? Helicase unwind the DNA coding for the protein. So unwinded means that they move the two strands away from each other. This means unwind. RNA polymerase will form the complementary strand. For example, in the first we have A, the complementary is T, C, G, G, C, A, T, and so on. So observe this document on document, for example, D, or for example, consider the sequence without the document. So consider the sequence of DNA molecule shown. Here we have the DNA molecule here, GCA, TGC, ATC, TGC, CAT, TG, and so on. So there's a lot. Here I stopped in this part. Here the sequence is called the non-template. Non-template is called the non-transcribed. If you watch the video, non-template is called the non-transcribed. It's the coding. So non-transcribed, non-template, it's the coding. So the sequence of the coding is similar to that of the mRNA, but we have to do something. How may you relate the mRNA molecule to the template to the non-template? Let's start first. This is the non-template strand. Non-template is called non-transcribed, it's called the coding. If we want to find the template strand, what should I do? I have to find the complementary strand. So complementary of G is C. Complementary of C is G. Complementary of A is T. T, A, G, C, C, G, A, T, T, A, G, C, and so on. So non-template is called non-transcribed, it's called the coding, while template is called transcribed. So you can simply TT, template, transcribed, non-coding. Non-T, non-template, non-transcribed, it's the coding. So the sequence of the mRNA is similar to that of the non-template, similar of the coding, but instead of T, it's replaced by U. Did you see here? I start only with the first three codons. Every three is called a codon. So here, these are called a codon. First codon, second codon, third codon, fourth codon, fifth codon, and so on. These are called nucleotide. First nucleotide, second nucleotide, third nucleotide, and so on. So the mRNA is the same, is identical, or is the same as the non-template. This is non-template, non-transcribed coding, but instead of T is replaced by U. Did you see here? I started with the first three. We have the same GCA as GCA. TGC, T here is replaced by U. ATC, T is replaced by U, so it becomes AUC. So mRNA, you have to know this and you have to study this. Keep it in your mind. mRNA strand is identical to the non-template. meaning that it's identical to the non-transcribed, meaning that it's identical to the coding, but the T is replaced by U, while mRNA is complementary to the template. Here, template transcribed non-coding. Template C, the complementary is G. G, the complementary is C. T, the complementary is A. A, the complementary is T. But we don't have to put T in the mRNA. We have to put U. C, G, G, C. T, the complementary is A. A, the complementary is T. But we don't have to put T in the mRNA. So in the mRNA, there is no T. You can solve it using two ways. First way, using the non-template, which is the non-transcribed, and it's the coding. Same sequence, instead of T is replaced by U. Or you can use the template, which is the transcribed, which is the non-coding, but you have to make it complementary. So, and you have to replace the T in the mRNA by U. So this is the function of the enzymes helicase and unwind the DNA. As I said, RNA polymerase synthesis the complementary strand. Translation, the second step of protein synthesis. The first step, it was the transcription. Second is the translation, where RNA goes for production of protein. Some of these protein will remain in the cell, others they are exported. So we have this sequence of the transcribed. If I ask transcribed, it's the template, it's the non-coding. Give the sequence of the transcribed strand, and here they gave the coding. Coding, it's non-transcribed, non-template coding, so it's non-transcribed. I have to give the transcribed strand. What do you have to do? Complementary. A, T, A, T, A, T, C, G, C, G, C, G, 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 it becomes C, C, C. C, A, A, the complementary of C is G, and the complementary of A, it's T. So it's become G, T, 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 C, G, A, G, C. Complementary. Specify the sequence of the mRNA. You have to specify the sequence of the mRNA coded. Sequence of the mRNA, either they use the coding instead of T is replaced by U, or you use the non-coding and you make it complementary. Here, transcribe, it's we have first AAA, it becomes AAA becomes TTT, CCC, GGG. We have GGG, it becomes CCC. CAA, it becomes GTT and AGC. Coding strand, here we have the coding strand. Okay, here is the coding strand. Coding is the same as mRNA, but instead of T is replaced by U. So first codon, the same. Second codon, the same. Third codon, the same. Fourth codon, also the same. But the fifth codon, TCG, will become UCG. Okay? 43, they said using the genetic code table, give the sequence of the amino acid produced. Here we have the amino acid according to the mRNA. AAA. How can I get it? Here, the genetic code table, this is given. Genetic code table, AAA. First letter here, first letter is A. Second letter, it's A. And the third letter is A. So A, move here, A, LYS. You have to study them? Sure, no. You have to revise them and you have to know every codon it codes for which amino acid? No. But you have to know only 3 of them. You have to know UAA, it's a stop codon, UAG, and UGA. Simply study UAA, one of them. UAG, you added G, and you added 2G. 2G, UGA, UAG. So you substitute the G with A. UAA, UAG, UGA. These are the stop codons. These 3 you have to study them. Okay?